Hi, everyone. I'm going to be giving my best Mr. Anderson impersonation today. So we are going to look at topic 4.6 for the cell cycle. So I kind of created like a graphic organizer, and so hopefully this can be a tool. This is optional to fill out, but these are some of the terms that we are going to be discussing in the notes today. And so I think this is a very visual topic. It's a very vocab heavy topic, but I think once you get the ideas of the vocab down, I think it's a very manageable unit that you apply to a lot of similarly worded or similarly challenged type questions. So this is here just as like an organizer if you want to fill out and as a way to kind of help you prepare as we get closer to our assessment. So we're just going to talk about the cell cycle today and basically how one cell becomes two. And so you might also see a lot of terms that might come up or be familiar from a review of when you were in biology as a freshman. And so the big idea for this topic is how does a cell divide to replace damaged or diseased cells? And so this is going to be a little bit different found in prokaryotes versus eukaryotes. So starting with prokaryotes, these are simple-celled organisms without a nucleus, and they actually contain their DNA. We know it's found in the cytoplasm, but they only have one chromosome, a single circular chromosome of DNA. And similar to eukaryotic cells, all organisms need to duplicate their DNA before they divide because we need to make sure or ensure that we have the same amount of DNA in both cells after division takes place. So the DNA gets duplicated, and in addition, prokaryotic cells, they have this other thing that stores genetic material. This is called a plasmid. Okay, and so a plasmid is also a circular DNA molecule. It's not nearly as big, but it's also found in the cytoplasm, and the plasmid will replicate independently. It is going to contain a small number of genes, and plasmids are most commonly found in bacteria, but they can also be present in a few eukaryotic organisms like yeast. Plasmids are going to be really interesting for us later on in a different unit when we look at taking genes out of plasmids and inserting other genes inside those plasmids and getting different gene expressions. This is an early idea or version of what we'll see for CRISPR or gene editing. And so for prokaryotes to divide, they just have to grow in size, right? They need to have the same nutrients, same genetic material, same organelles in both cells. So they start to double in size. And for prokaryotes, this is just called binary fission. Okay, and it's a very simple process. There's not a lot we're going to go into detail for this. If we take a look at this diagram, we can see the plasmids, we can see the chromosomes, or the single circular chromosome, and then over here we have ribosomes. And for, again, cell division, the circular chromosomes will start to replicate at what we call the point of origin, or the origin of replication. And eventually, once everything has sufficient nutrients and sufficient size, the cell becomes two. Again, that's specific to binary fission, or binary fission is specific to prokaryotes. And so the bulk of this unit, or this topic, is found in cell division in eukaryotic cells. So how do our cells become two? And so we know the cells will spend their lifespan going through the cell cycle. And so this is a series of steps where it's preparing for division and eventually will divide. And so there's two general steps. I will say too, as we go through this, this is going to be a very important diagram. You definitely need to know this, and you need to know where things are. I would highlight the M phase here. For example, there's going to be a lot of questions related to the M phase in the AP Classroom questions. So we have to make sure we know what's going on in which phases and what they are doing. All right, this is where the vocab comes into play. And so the two main phases of the cell cycle, they are interphase and cell division. Okay, and so we'll talk about interphase first. Interphase is basically all about growing and preparing for division. And so this is where where cells spend most of their time. This is quote unquote the normal state. And so they have their nucleus, they have their organelles, they're not actively dividing at all, but they're basically just getting ready for division to take place. And there's three steps within interphase. You might remember these. These are G1, where the cell is growing. There is the S phase. S stands for synthesis, and this is going to be an important topic here. And then we'll circle back to that here in a second. Our last step is G2. So the S phase is going to be important because we have to duplicate all of our DNA, kind of what I mentioned earlier. And so this needs to happen, and cells need to make sure the DNA gets duplicated. If not, the cells duplicate with an unequal amount of DNA, and that can lead to mutations. That can lead to the cells just not being able to be viable or survive and just break it down and die. And so what we say here, this is kind of a big idea. For mitosis, or for cell division, we go from 4 unduplicated chromosomes to 46 unduplicated chromosomes to 46 duplicated chromosomes. And what we'll see here is that DNA and genetic material can be stored in a few different ways inside of a cell, depending on where it is in the cell cycle. For interphase, cells generally keep their DNA in the form of chromatin. So we'll talk about that here in a second. But just going back to this idea, unduplicated to duplicated. So our chromosome number has not changed because they are attached to each other. So here we have 2 unduplicated chromosomes, and then as the S phase occurs, we would say we have 2 duplicated chromosomes. And so these duplicated chromosomes are held together by this structure called the centromere, and these are also called sister chromatids. And so what happens during the S phase is the chromosome number doesn't change, right? We go from 46 to 46. However, our DNA has doubled. So we go from, if this is like 100% of our DNA, we go to 200%. Okay, because we obviously we can see there's just more things here. There's more physical material. And so the DNA changes, but the chromosomes do not. And that's where the 46 unduplicated to 46 duplicated comes into play. And then the last step, the G2 phase, basically the cell continues to grow. The cytoplasm needs to basically double all of its components, and that includes like organelles. So organelles are going to be duplicated here as well. We talked before, like chloroplasts and mitochondria, for example, they used to be independent organisms. They divide independent of the rest of the cell because they have their own DNA. And so they divide. Once the cells have sufficient amount of mitochondria or chloroplasts or both, then the cell is able to continue its process of cell division. And so kind of taking a look inside the nucleus during interphase, we have DNA, and that's going to be found in the form of chromatin. The best way I think about chromatin is kind of like just like a bowl of spaghetti. It's unaccessible. It has genes in there. But the cell can't or won't be able to duplicate it until it condenses. And so chromatin is going to condense into chromosomes. Okay, and those are the X's that we're probably pretty familiar with. And the DNA, chromatin is made up of 2 things. It's made up of DNA wrapped around these proteins called histones. And so DNA wrapped around histones, together that makes up what we call the nucleosome. Okay, so let's take a look at some of these terms. So the histones are these purple spheres here, and the DNA is kind of like a wire. And so the histones are there so the DNA are more organized, and so they take up less space. You think about like a phone line that has those squiggly lines, right? That's so they can kind of like coil up and take up less space. And so this will make up a nucleosome. So if we go over here, again, we have our histones, we have our DNA. This is one single nucleosome. And all chromatin is just repeating nucleosomes kind of clumped up together that will form or make up all of our genetic material inside our nucleus. So that's basically it for interphase. Now we get to like the more action part of the cell cycle, which is cell division. super important to know. Cell division is also known as the M phase. This is not just talking about mitosis. This is talking about mitosis and cytokinesis. So it sounds kind of like semantical, but basically M does stand for mitosis. However, cytokinesis is still a part of that as well. So all of cell division is the M phase. Mitosis does not complete cell division, right? It just will eventually, what we'll see is that it divides the nucleus. And so that's what we'll talk about first, is how the nucleus in the genetic material divides and duplicates, or just divides, it's already duplicated in the S phase, and how that is transferred to 2 new cells. And so this is where maybe some of the review might start kicking in, but we have 4 phases in mitosis. This is called PMAT, right? Prophase, metaphase, anaphase, telophase. So we'll kind of explore what's going on in this model. All right, and so we start with prophase, and this is where I would say this is probably where the most stuff is going on. This is the first step of mitosis. We basically have to just like start getting all of this vocab down. So in prophase, the nuclear envelope needs to break down, right? We need to be able to move our eventually chromosomes around to different sides of the cell. The chromatin, right, our bowl of spaghetti, is condensing into chromosomes. Again, X-shaped. And these are connecting, the chromosomes are connecting the sister chromatids by the centromere. So the centromere, you can see here, it's connecting them in the middle. And then there's a very similar sounding word, it makes it kind of extra confusing. There are centrosomes, which are composed of 2 centrioles. So they are just trying to create as many similar sounding words as possible. So you got centrioles over here on the side. These are going to be, you can see microtubules here. This is where eventually spindle fibers will come out of, which is our next term. And so spindle fibers start to form, and they're using these centrioles as like anchors. So eventually we'll see like that they like form spindle fibers out of the centrioles together, which make up the centrosome, eventually to grab those chromosomes. Okay, so there are 4 phases of mitosis, but there's kind of a 1.5 step. This is prometaphase. And so during prometaphase, it's kind of the in-between between the two. Microtubules from centrosomes move towards the chromosomes. So we can start to see that here, right? This is our centrosome. Here we can kind of see 2 centrioles, and our spindle fibers are coming out of these centrioles and eventually attaching to the chromosomes, and they attach to the chromosomes at the centrosome, but this is another word, or the centromere, excuse me. See, I even mixed them up. But at the centromere, we have this actual physical thing called the kinetochore. Kinetochore. Okay, and so the kinetochore is a protein found at the centromere, and this is where the spindle fibers will attach themselves to chromosomes to eventually, they're going to yank them out, or yank them to opposite sides. And so the kinetochore is in pink there, spindle fibers attaching to the kinetochore. Kinetochore is found at the centromere, which also connects the 2 sister chromatids, which makes up 1 duplicated chromosome. So again, a lot of vocab. So the visuals, studying this is very much needed to have a good grasp of these concepts. But now that the chromosomes have been attached to by these spindle fibers, we can start moving them around. And so this is where metaphase is. Metaphase is very short and hopefully pretty easy to remember. Metaphase is all about chromosomes lining up along the middle metaphase plate, which is called the middle part or the middle part of the cell, right? So M, metaphase, middle. All right, and so now we can see during metaphase that our cells, or within the cell, our chromosomes are all along the middle. And so that takes us to our exciting part, separating. So this is anaphase. So anaphase is where there's some big ideas coming up. This is where the duplicated chromosomes get pulled apart to opposite sides, to centrosomes, right? So our centrosomes, again, these are our centrosomes. And so what happens is we go from duplicated chromosomes to one duplicated chromosome to two unduplicated chromosomes as soon as sister chromatids separate. So there's actually this is very kind of like semantical, but I actually want to update this. Mitosis goes from 46 chromosomes to, we actually would say like, you say like 46 unduplicated, or we could say like 92, which is just double 92 chromosomes. Because this is one chromosome. This is one duplicated chromosome. And then as soon as they split, we say this is now two chromosomes. Okay, they're sister chromatids, but now they're also chromosomes by themselves. Because now they're no longer duplicated, or they no longer share, or they will be part of different cells. So they are individual chromosomes. So that's why we say, and that's why I had that typo, we go from 46 to 92. It's still within one cell. Eventually, that will be two. Okay, and so that takes us to our last step of mitosis, which is telophase. Telophase is all about basically repeating or undoing all of those steps in prophase, except we have two cells now. We're about to have two cells. So where we had a nuclear envelope break down, here we have two daughter nuclei start to form. The chromosomes become less condensed, right? So we can kind of see our sister chromatids, which are our chromosomes. They are going to decondense, eventually forming our chromatin. So now we're basically putting cells back into interphase here in a second. The remaining spindle microtubules that were used to move those chromosomes to opposite sides are broken down, and mitosis is complete. Nice. Okay, so we still don't technically have two cells yet, though, right? We have two nuclei, one cell. And so we still have to basically separate the cytoplasms of the two cells to officially or actually make them two. And so this is called cytokinesis. All right, if we break that word down, right, cyto, we know this is about like cell. This means cell. Kinesis is just like movement. And so this is like moving a cell to two different, moving the cytoplasm to two different cells. And so this ensures that equal distribution of cytoplasms get into both daughter cells, so they have both of the nutrients that they need to survive. And once cytokinesis happens, there is this it's outside of the cell cycle kind of, it's called G0, which we just have G0. And this is, if we could scroll back up real quick, we can kind of see G0 over here. It's basically like cells getting off of the road, off to the side of the road, and are just kind of like maintaining stability. They're not going through the process of dividing, they're not growing, they're not doing anything. It's just like a healthy state for cells to be in. And this is the state a lot of cells want to be in. Maybe they don't want to always actively be divided. Most cells in our body aren't actively dividing and are in G0 phase. And then whenever there are certain signals, and this gets into topic 4.7, certain signals can induce cell division and can start cell division. And sometimes there are errors in those signals, and that can lead to cells dividing uncontrollably, which is going to lead us to talk about what cancer cells are and how cancer can form from deregulating the cell cycle. And so in animal cells, cytokinesis involves the formation. It's different in animal cells and plant cells. In animal cells, it's called a cleavage furrow, which just pinches the cell in two. So I always say it kind of looks like a peanut, but it kind of pinches. So there's a cleavage furrow, pinches, and then eventually we'll separate them in an animal cell, right? In plant cells, they have to deal with the cell wall, right? They can't do that. They're not as fluid or bendable as an animal cell is. And so they have to form basically like a new cell wall in between their two nuclei. So for plant cells, that means they have to form a cell plate. And so that will form in the dividing cells. You can see like Golgi vesicles. So Golgi vesicles are like transporting cell plates. If you remember, cell plates or cell walls are made out of this cellulose, which is a macromolecule. It's like a carbohydrate. So it's good for rigidity and keeping plants upright. And eventually that cell plate will form two separate cell walls for two complete plant cells. And that's it. So one cell have become two. Again, there's a lot of vocab we want to talk about. Some of those terms in the table we can talk about at a later topic. Here's an overview for mitosis, and I hope that was helpful.